So we've got the mucosa, which is the inner lining of the lumen. We got the submucosa, which is mainly connective tissue. The muscularis externa, mainly smooth muscle, and then the serosa, another connective tissue layer. So let's talk about the mucosa first off. It's separate from the GI, right? It's going to help determine internal body environment from external body environments. It's basically the lining of the GI lumen itself. These cells that make up the epithelial lining here, we call them enterocytes. There are different types of cells that have different functions. We've got cells that are in charge of absorbing. These are absorptive cells. We've got exocrine cells, which secrete usually glandular secretions or digestive juices. We've got endocrine cells that are secreting hormones that are all a part of the enterocyte or that lining epithelium. Now we talked about the subdivisions of the mucosa, which are going to be the cells, the epithelia. Then we've got the connective tissue layer, which is the lamina propria. And just to remind ourselves here, the epithelia doesn't have blood vessels, and we talked about that in our last semester. Epithelia, that lining of cells, do not have their own blood supply, and so in order for them to get nutrients and oxygen, there must be an underlying lamina propria. So that's the reason we've got this lamina propria, which is a connective tissue layer that has the small vessels and small nerves that are actually feeding and innervating the epithelial layer, because the epithelia itself is avascular. And then finally, we've got the muscularis mucosa, which is a thin layer of smooth muscle that is a part of that mucosa. Now, the function of the muscularis mucosa is to help contract the mucosa. That's going to be really important for moving food along the lumen. So that small layer of smooth muscle is really contracting to help move fluid, move food along the lumen of the GI, okay? Now, underneath that, we've got the lamina propria. So the lamina propria is the second layer of the mucosa. This is a connective tissue layer. Again, this is pretty much repetition. We've got small vessels here, which are helping to feed the epithelial layer. We also have certain lymphatic tissues, such as lymph nodes or nodules, and then Peyer's patches are another aggregate of lymphatic tissue. Peyer's patches are only found in the ileum, which is the distal most part of the small intestine. So I'll talk about unique features to the small intestines, and Peyer's patches is one of those. So you're not going to find it everywhere throughout the GI tract, but only in the ileum. And then finally, the muscularis mucosae, we said, is that thin layer of smooth muscle. It's going to be longitudinal fibers, as well as some circular fibers. So there's going to be a difference in the direction of the fibers here. And again, that is helping to mix or move the contents of the lumen. So we've got that small strip of smooth muscle with fibers going in alternate directions so that we can squeeze the GI tube horizontally, but we can also squeeze it longitudinally and really mix the contents of the lumen very well. Oops, my phone's ringing. Okay. All right. Then we get to the submucosa, which is that second layer. This is a thicker layer of connective tissue. This layer is more in charge of distensibility, elasticity, structural support of the GI tract. This is going to have larger blood vessels, which are coming in from the mesentery, as well as the larger nerves that are forming the submucosal plexus here. This plexus we described as being the Meissner's plexus, which is a part of the enteric nervous system. Now, when we say enteric nervous system, this is really meaning the autonomic branch of the nervous system, where we have mainly the parasympathetic innervation of the GI tract. Okay, so this is the second layer out from the mucosa. Then we get the muscularis externa, again, a thicker layer of smooth muscle. This is also arranged in alternate directions. So the inner layer has circular fibers, which help to change the diameter of the tube. The outer layer has longitudinal fibers, which help to change the length of the tube, right? So we have fibers that are contracting, making the lumen smaller, fibers that are contracting, stretching the length of the tube, which is contributing to really mixing or mulching, we can say, the contents of the lumen of the GI. We also have another plexus here, the myenteric plexus, sometimes called the Auerbach's plexus, which is a second part of that enteric nervous system as well. And then finally, the serosa, which is the outermost layer that has two components to it, an inner connective tissue layer, and then an outer epithelial tissue layer, which we call the mesothelium. The mesothelium is sometimes called a serous membrane, and that's what I mean when I talk about that very thin layer, which is continuous with the mesentery or continuous with the peritoneum. So here we can see that connection. And if you remember from lab, we talked about the mesentery. And the mesentery is where we have the connection that's coming from the major vessels, so coming from the aorta, from the vena cava, and bringing those vessels those vessels into the GI structures themselves. So the mesentery is the connection that contains the nerves and arteries and veins that are coming to the GI tract. Okay, so that's pretty much the structure of the GI. We're going to move on to look at the functionality of each of the parts, each of the components, and really look at what role each part of the GI is playing in the overall function of digestion and absorption. So we got the mouth, the pharynx, the esophagus. We've got the stomach, which is really in charge of mixing, breaking down parts of the food particles or the macromolecules. Remember, in the stomach, we got this really acidic environment that's helping to break down proteins and fats, starting the breakdown process. We've got the small intestines where that digestion is further continued, and then we begin absorption, right? The main role of the small intestines is absorption. That's followed by the colon. The main role of the colon is really condensation of the waste, reabsorbing most of the water from the contents, and then solidification of that to be expelled through the rectum and anus. So digestion really begins in the mouth. It begins with a process called mastication, which means chewing of our larger food particles and beginning the breakdown process, breaking these down into smaller food particles. Things like carbohydrates begin to digest in the mouth. There are certain enzymes in our saliva that start breaking down carbohydrates in the mouth. Saliva is also going to be important for lubricating the oral cavity and beginning lubrication of the esophagus as that food moves through the upper GI tract. We've also got salivary amylase. That is the digestive enzyme that breaks down starch specifically, as well as glycogen in the mouth. This is why starchy foods or carbohydrate-rich foods, usually we can begin to taste that sweetness in the mouth as that salivary amylase begins breaking down starches and carbs in the mouth. Then that leads us to the pharynx. The pharynx is pretty much the open way or the passageway into the esophagus. This is what we commonly call the throat. Remember, this is a common passageway, not just for food, but also air. This is a shared passageway between the larynx, which is the opening into the trachea. So this brings up the question of making sure that food is going into the right passageway and not going into the larynx, which is going towards the bronchus and the airway. Beyond that, we enter the esophagus. So the esophagus is the opening down into the GI tract, so to speak. It's a muscular hollow tube. It's going from the pharynx to the stomach. It's going to be mixed in terms of the type of muscle that's making up the esophagus. We got the upper 1/3 formed by skeletal muscle, the lower 2/3 formed by smooth muscle. And right away, that should tell us about the control that we have over different parts of the esophagus. So the upper 1/3 we can control, right? It's skeletal muscle, so we have voluntary control over that part of the esophagus. We also have voluntary control over the upper esophageal sphincter. So this is a strip of smooth muscle that controls the opening from the pharynx into the esophagus. And because that is made of skeletal muscle, we actually have voluntary control over the upper third as well as the upper esophageal sphincter. And when I say we have voluntary control, this is what I mean by, this is what allows us rather to be able to swallow intentionally. So we can decide to swallow, we can decide to move food from the pharynx into the esophagus. We have voluntary control over that bit. The lower 2/3 of the esophagus is smooth muscle. So we do not have voluntary control over this part of the GI, this part of the esophagus, as well as the lower esophageal sphincter. Again, there's another sphincter in between the end of the esophagus and the opening into the stomach that is made of smooth muscle that regulates the emptying of the esophagus into the stomach. Because it's made of smooth muscle, we do not have control over this. And again, because the lower 2/3 is made of smooth muscle, we cannot regurgitate food, we cannot control any aspect of the movement of food beyond the upper third of the esophagus. Okay, so really interesting dynamic here of how the esophagus is a shared structure. Parts of it we can control and parts of it we cannot.